wherever you might find yourself.
Oh, I think if it's not just me, I think we've actually lost Elvis for a second there.
Hello?
Fedora, is that correct?
Oh, can I go ahead? I think we lost Elvis there.
Yeah, sorry. I think I was taken out.
We'll
so
wait
I'm
for
back
him
now.
to either join or
I'm here.
either join.
I think
Okay,
yeah, I was
Elvis.
taken out.
All right.
Yeah. Sorry.
welcome
Hello,
back.
everyone. Thank you for joining us today on a Pan Microplast Club Talk 4. Today we'll be hearing from Dr. Oluni Fadari on the topic Aquatic Ecotoxicity of Microplastics, Perspectives from Animal Models.
All right, can I go ahead?
Dr. Oluni Fadari is a research fellow at the Center for Energy Research and Development, Obafemi Awolowo University, Il-Ife. He's a coordinator of the Pan-African Microplastic Research Network and a member of the Chinese Association of Scientists, Young Scientists for Sustainable Development Goals. Dr. Fadare is one of the young African scientists involved in microplastic research in the region, with specialty in analytical environmental chemistry and ecotoxicology. He employs spectroscopic techniques and molecular assays to understand the behavior and effect of microplastics in different environmental matrices and potential impact on humans, involved in advocacy, policy formulation, and public enlightenment on circular economy of single-use plastics. I'll hand over to Dr. Fadari to take us through his presentation. Thank you.
Thanks very much, Elvis. Good day, everyone. I hope we can all hear me. I will be presenting this afternoon on the topic Aquatic Ecotoxicity of Micro Nanoplastics from the perspective of animal models. Let me say that there are so many studies that have been conducted, at least in the last 5-6 years, on microplastics distribution and occurrence in different environmental compartments. However, little studies have emerged in the area of the toxicological study of microplastics. And you agree with me that without the toxicological study, it may be difficult to assess the risk of microplastics to human. And that's what I'm going to be sharing some of my experience in this area of using animal models to predict micro and nanoplastic toxicity. Okay, let me introduce my center. As you can see here, I had my PhD at the Research Center for Eco-Environmental Sciences, about 9,900 kilometers away from Nigeria, where I was also research assistant. But because of COVID, I have to come back to Nigeria. And for now, my engagement in Research Center of Eco-Environmental Sciences is on suspension. So, but presently I work with the Center for Energy Research and Development. And this is located in Obafemi Awolowo University, as you've heard in the introduction. The university itself was created in 1961, while the center was created in 1976. The center has 5 divisions, and one of which is environmental and earth sciences, where I work presently. Yeah, by the introduction, this just shows the distribution of plastic production globally. This is 2019 report. Asia is the largest producer of plastic products across the world, with China being the leading country, producing more than 50% of what the whole Asia is producing. We can see Middle East and Africa here producing a very small percentage of about 7%. But this does not imply that this is all what is available in Africa, because from these richer countries or the high producing plastic countries, they are also imported into Africa, and through maybe packaging materials and also plastic products are imported from these countries. So despite the fact that we do not produce much plastic, we import some of these plastic products into the region. And from here, I show some of the plastic waste. Apart from the plastic products, you know that there's been some agitation of recent. I think United Kingdom just passed a law restricting or prohibiting importation of mismanaged plastic or used plastic, as it may be, into low-income countries. And presently, China produced the highest plastic, while United States is the highest producer of mismanaged plastic, as it were, according to the recent report that was published in Nature. And as of 2019, the total production of plastic stand at 370 million metric tons. But you agree with me that with the emergence of COVID, we have something that is more higher than that now. And this is also a huge global market, which is to the tune of 554 million US dollars as of 2019. And it's projected to rise up to like 720 million US dollars by 2025. Yeah, I just put here the process by which microplastics degrade in the environment using different mediums. This picture was captured at the beach of the Gulf of Guinea in southwestern Nigeria, January 2021. You can see the environmental degradation of plastic products along the beach. You know, we have what we call primary nanoplastics, which are introduced into our different personal care products. Here, I show different products that has been reported to contain microplastics. We can see that even the air we breathe in also contain different types and shapes of microplastics. A study was conducted of recent, I think in Elvis lab, if I'm right, in Australia, where they identify isolated microplastics from rice. And I think about 3 grams, if I'm right, is estimated per bag, if I can recollect that study. From the seafood, a lot of microplastics have been isolated. A study conducted in the United States estimated that about 37,000 microplastic is consumed per year for anyone that is source of drinking water is from the bottled water. And all this, as you know, we've had 1 or 2 cases of presence of microplastic in almost all the items that's highlighted. And the worrisome aspect of it, of course, microplastic has also been detected in humans too. And earlier this year, a report from Italy shows that microplastics can actually be transferred from mother to the child, as a study isolated microplastics from woman placenta. And it was discovered in that study that 2 out of every 4 placenta actually contain microplastics. Yeah, I put that why should we care about microplastic? If there is microplastic everywhere, what is the implication? I'm sure some of us will have attended conferences whereby we discuss the presence of microplastics, you know, in different compartments. And you know, most of the questions that always come up is, and so what? Air is also present everywhere. Water is present everywhere. So what is the implication? And I did a rough search using the Google Scholar to see the studies that have been conducted on the occurrence of microplastics, and as well as the toxicological studies. As you can see here, we have just about 32% of toxicological studies that have been carried out on different aquatic animals. And no study up to date, clinical study, has been carried out on human. from different environmental compartments, we have more than 68% of studies that have examined microplastics in different environments. So I could say that there is a need to increase our knowledge, the global knowledge on microplastic effects on the environment. In that, yes, there's no doubt that microplastic is present almost everywhere now, but there's a need to increase our knowledge. We need to improve our knowledge beyond the presence of microplastics everywhere. Then we have to move from that information to understand the risk it poses to the environment as well as human. And we have the toxicological mechanisms that has been established. We have the physical and the mechanical toxicity. We have the chemical and the biological. The physical is injection of plastic products, immobility, blockage of digestive system, suffocation, and the rest. That shows the physical toxicity. In the chemical toxicity, it has been proven that microplastic acts as vectors for persistent organic pollutants in the environment. And also at the biological level, we talk about the oxidative stress. We talk about inhibition of radical scavenging agents, the inflammation of organs and the likes. Yeah, I put the different method that's been used in toxicological study of microplastics. We talk about the cell toxicity assay, in which different cell lines are subjected to microplastics of different kinds. And the cell viability is examined over a period of time or at a certain time. We have the acute or chronic toxicity assay. We have the gene expression, that's gene transcription. We have the fluorescent imaging and also the protometric technique. And I will be discussing some of these techniques as regards to animal models. Yes, some of these studies have been predicted and some has been established in different organisms. But in literature, we don't have enough studies yet that can really help to determine the risk assessment of microplastic, both to human and to animals in the environment. Now, I want to move very fast because I still have so much to cover. In some of the studies that were conducted in our lab, we tried to answer the question, are microplastic toxic? And you agree with me that in different fora, people will have asked that question. You're talking about microplastic almost everywhere. Is it toxic? What is the effect? So in answering this question, we used two animal models, Daphnia magna and Danio rerio. Daphnia magna is a water filter, it's a filter feeder. That is, it sources food from fresh water. By filtering the fresh water, it collects the particles there as food. And another reason why we use it is that Daphnia magna is known to be completely sequenced. That is, its genome is completely known. And also, it has a very short lifespan, which can actually help us to understand the life cycle of the effect of microplastics on this organism. And it can also replicate a lot of its offspring within a very short period of time. It takes between 5 and 7 days for it to reproduce its offspring. And in case of Danio rerio, it's a very transparent fish that you can actually examine what is going on with each of the organs by mere observing it under the microscope. And also, it also produces a large quantity which can be used also for study. And as well, it is known that about 68% of the genome of Danio rerio is also similar to what is obtainable in human. And in also understanding the toxicity of plastics, we know that pristine plastic does not exist in the environment. It has to interact with other environmental factors. So we decided to put this into consideration, in our experiment by looking at the humic substances, which is also very common in the environment, and also organic pollutants using dichlorodiphenyl trichloroethane as a model. Here I show the acute toxicity assay that was conducted using two different microplastics, 1 micron and 0.1 micron. And we subject Daphnia magna to this assay within 96 hours. And from this study, we can see that 1 micron after 96 hours at 400 milligram per liter, there is no mortality. It is 100% survival. And that shows that the size particle 1 micron does not have any toxicity effect on Daphnia magna. But in the case of 0.1 micron, we can see that with increase in time, that is decrease in percentage survival. And as at 72 hours for 200 milligram and 96 hours, for 72 hours, yes, for 200 milligram, and after 96 hours of 400 milligram, there is complete 100% mortality of the animals. Yes, we now try to look at what is the effect of humic substances if it's present when we introduce the nanoplastic. Of course, we concentrate on the nanoplastic, having understand that the microplastic, which is 1 micron, does not have any toxicity effect on the animal. We decided to concentrate on the 0.1 micron. And here, this is trying to subject the animal to nanoplastic in the presence of 3 different types of humic acid: the natural organic matter, the fulvic acid, and humic acid. And as you can see, there is a great reduction in the mortality of the animals. That, for instance, the humic acid, which has the highest survival, at 96 hours, we have about 70% survival compared to at 72 hours that we have 100% mortality in the nanoplastic. So we want to understand the toxicological mechanism of this toxicity in Daphnia magna. And we introduce a labeled microplastics, which can fluoresce under the lesser confocal microscope. And we expose Daphnia magna to this 0.1 micron and also 1 micron to understand the toxicological mechanism of the nanoplastic in Daphnia magna. And as we can see here, we can see the nanoparticles trapped within the carapace of the animal. And as this is not within the animal, it's within the feather that Daphnia magna used to filter its food, which means it cannot absorb the 0.1, and they are trapped within the carapace, sticking into the layer, the internal layer of the carapace. But in case of 1 micron, we can see that it can easily eject it and also expel it from the body of the animal. And this clearly shows a mechanism that a physical toxicity has occurred here, in that the nanoparticle, being small, are trapped on the surface of the carapace of the animal, thereby immobilizing the animal, and as a result, mortality occurs over time. We also want to understand the effect of humic acid. So we decided to introduce humic acid, into the exposure system. And from here, we use 2 concentrations, 100 milligram and 400 milligram. And from here, you can see that, just as I've explained the other time, the animal, actually was unable, to consume the nanoparticle. And even at 400 milligram of higher concentration, you can see that it's well trapped within the layers of the animal. But when 5 milligram per liter of humic acid was introduced, we can see in the gut of the animal that it was able to pick up the nanoplastic that it was unable to actually inject initially. And at a higher concentration of 400 milligram, you can actually see that it entered completely into the system of the animal and, trying to expel it. Compared to that of the 1 micron, in which at lower concentration, of course, you can see it in the gut, and at 400 milligram of a higher concentration, it is duly consumed without any of it being trapped within the carapace of the animal. Yes, this is gene expression. We try to look at the genes that are responsible for oxidative stress in the animal. We use 4 of them: the GST, the CAT, HSP70, and PGP. And just from this, you can see, and we compare it with the control, and in comparison with the control, we can see that truly the nanoplastic actually induced oxidative stress in the animal, but also in the presence of humic acid. But when we have the nanoplastic and the humic acid combined together, we can say that is drastic reduction and induction of oxidation across the 4 genes that were examined. So we concluded that nanoplastic is toxic and is consistent with what has been reported in literature. But in the presence of humic substances, which is actually present in the real environmental scenario, then it reduces the mortality rate in Daphnia magna. And humic acid displayed a detoxifying property in this experiment and this is attributed to what we refer to as corona formation. I'll be talking more about this later, and we discovered that, of course, the presence of humic substances actually dictate the behavior and the toxicity of nanoplastic in Daphnia magna. Away from that, the concept of corona formation, when microplastic or nanoplastic enter into an organic macromolecule, the macromolecule contests for the hydrophobic sites on the surface of the particle, thereby form what you call corona. And we have 2 types, what we call the ecocorona and the protein corona. The ecocorona are the macromolecules that pick up from the environment, while the protein corona are the ones that are picked up from the physiological fluid of the animal, whether the blood, whether the serum, or any other physiological fluid. And Jean et al. Actually expressed this in 2013, talking about the process that takes place, the interaction that takes place between a nanoparticle and that of protein. That can be conformational change in the protein. The activity of the protein can change when it's come in contact with the particles. Then we have what we call the soft corona and the hard corona. The soft corona are the one that can easily be detached from the surface of the particle and be replaced by another. But the hard corona is actually referred to as the fingerprint on the particle, that once it's attached to it, it's actually difficult to remove it from the surface of the particle. And this can actually be used to estimate or predict the toxicity that such particles can pose to organisms. So what we do in trying to understand this is that we subjected Daphnia magna secreted protein. The secreted protein in this sense is that we collected the neonates that is less than 24 hours and we put them in a separate medium for 48 hours. And the secreted metabolic macromolecules were collected while we homogenized the organism. And we now incubated it with the nanoparticle, also in the presence of humic substances. And we have to centrifuge it in order to precipitate the particles, also to reduce an alkylation for the protein denaturalization. Then we subject it to separation using gel electrophoresis, and we also do the protometric analysis using SC mass. Here, this is the SDS page electrophoresis showing different bands of the protein that was separated from the protein corona, and the second one from the ecocorona. We decided to concentrate on the high abundance proteins. We concentrated on the high abundance protein in the 2 corona. And from the study, we realized that we're able to identify 281 proteins from the ecocorona, and from the protein corona, we identified about 723 proteins. And we have about 106 similar to both corona. So this is the protometric analysis results showing some of the proteins that were identified. And interestingly, this is about 9 proteins. In checking with the literature, we discovered that they are actually responsible for the immune defense, the cell maintenance, and anti-predatory response of Daphnia magna in the environment. And by implication, they also are responsible for cell signaling and communication in the aquatic medium. And the implication of this is that whenever microplastics get into the aquatic medium where you have Daphnia magna, once these proteins are absorbed, it disturbs the communication of the animal, within its habitat, and that can actually expose the animal to predators. And that is one of the interesting findings from this protometric result. This for the ecocorona, and for the protein corona, we identify 3 distinct proteins, the apolipophorins and the superdioxide dismutase and the vitellogene. These 3 proteins are also important in the lipid transportation, the antioxidant effect, as well as the estrogen mediation of the Daphnia magna. And the implication of this is this: for the lipid transportation, it means that the metabolic activity of the animal will definitely be reduced, or be affected. And for the superoxidase dismutase, we are talking about the reduction in the activity of the antioxidant agents, that is the enzymes that are responsible to which we are called the radical scavengers, will also be, the activity will be reduced. And for the vitellogenes, that is more responsible for the reproduction, the estrogen disruption. So the implication is that a lot of literatures has actually reported some of this, but none has actually been able to prove the mechanism by which this occurs. And this is what we have been able to do using protometric analysis. So away from that, I want to also talk about the effect of nanoplastics with organic pollutants. And in this case, we use zebrafish larvae to carry out this study. And what we did was that we introduced nanoplastic in different concentration to zebrafish embryo, the fertilized embryo, but up to 96 hours, there is 100% survival. So it was not reported, I didn't show it here, but there is no deaths throughout the experimental period. We know, we have the knowledge that DDT can actually induce a neurotoxic effect, it can have toxicity effect on the zebrafish embryo based on previous studies. So, and as you can see here, with increase in concentration at time, of course, there's also increase in mortality when we introduce the DDT. But when DDT and MPP were introduced, we observe a greater mortality in the animal. So in the 96 hours, L50 of the DDT is about 7.73 milligram per liter. But this decreased sharply when we introduce nanoplastic into about 2.13 milligram per liter, which shows that although nanoplastic is not toxic to the zebrafish embryo, but is acting as what we call a potentiating agent for DDT. And in order to understand this, we also carry out the bioimaging, the fluorescent bioimaging, trying to understand what is the behavior of the nanoplastic with the animal. And our observation is that as the increase in concentration of the nanoplastic to zebrafish, we can see that there is more of these particles that are absorbed on the surface of the larvae. And the implication is that when nanoplastic absorb DDT on the surface and is now sticking to the body of the animal, the more of the DDT are introduced into the animal, more than when is only DDT that is present. And this actually explains the effect of the potentiating effect that we initially observed in nanoplastic. Now we quantify it using fluorescence method, and we discovered that with increase in time, of course, there is increase in intensity. Yes, we also look at the neurotoxic effects of MPP and DDT. And just as we can see in this graph, the DDT is indicated. We look at the inhibition of the acetylcholinesterase activity, the ACHE activity in zebrafish larvae, and discover that in the presence of MPP, there is more reduction in the activity of the ACHE than when DDT is present, which shows that in the presence of MPP, it's still establishing the same fact that in the presence of MPP, DDT become more toxic to zebrafish larvae. And lastly, we look at the genes that are responsible for neurotoxicity and also hepatotoxicity. That's for liver activity, which is more of metabolic activity of the larvae. And we also look at the ones that is responsible for the the reproductive effect. That's the vitilo gene, which is estrogen disruption. And if you look at all these figures, they look very similar. And the implication is that just as we have seen in other studies that have shown, in the presence of MPP, DDT induced more of these genes, which shows higher toxicity, of DDT in the presence of nanoplastic to zebrafish larvae. And this is very important and fundamental to toxicological study. And we understand that there is no effect of MPP when zebrafish larvae is exposed to it. But in the presence of DDT, it induced more toxicity, which shows a potentiating effect of MPP in zebrafish. And this simply explains that although nanoplastic may not be toxic to this organism that we have examined, but when it coexists with other organic pollutants in the environment, it can actually induce more toxicity. Yeah, in conclusion, I put it that effective policy of formulation can only be possible when scientists focus more on providing toxicological evidence of micro nanoplastic effects on the environment and on human health. And secondly, toxicological mechanism is important because this will help us to mitigate the adverse effects of microplastic on public health and the environment. This is my research team in Beijing, under the able leadership of Professor Liang Guo. And these are my wonderful research members. These are some of the selected publications. Thank you very much for your attention.
Thank you, Fadara, for such a wonderful talk and amazing study you've done in there. I think for now, we'll open the floor for questions from people who think they have questions for Fadara. I don't know, Holly, because I'm using the phone, I don't know if you can help with, I don't know if someone raised their hand, if I can see from here. But I think if you have a question, you can unmute yourself and just, yeah.
I think.
Thank you.
Is Andrews raising his hand?
Hi, I don't think my video is working, but hopefully you can hear me. Thanks very much for that really interesting talk. I wondered if you could say a bit more about the nature of your 0.1 micron nanoplastics. What are they? What's their surface charge? And did you look at different types? Because we've done, well, quite a long time ago now in a completely different context. We did quite a lot of work on this and found that it's very, very dependent on the surface properties of the polymer particles. So I just wondered if you could comment on that, please.
Yeah, because thank you very much for the question. I didn't put the detail there because I'm trying to work within time. Actually, we use polystyrene nanoparticles and being produced from we purchased it from the commercial industry, and we have the NH2 as the surface charge. Is an amide group, for the surface charge, and the size. We also establish the size to understand that we also establish what the manufacturers has put there using nanosizer. And I think the what's it called now? The size for the 1 micron is about between 1 micron and about 1.2 micron range. While the 0.1 micron is about 80 to 100 nanometer. I don't know whether I've answered the question.
Mm-hmm. Well, it's interesting that they're amine coated because polyamines are known to have quite a lot of cellular effects and possibly cytotoxic effects in drug delivery and all sorts of things. But I'm also interested in the fact that they're polystyrene, which is normally considered to be very hydrophobic.
Yeah.
But obviously, once you get a corona on it, it might not be so hydrophobic. But I guess the interesting thing about it is that it attracts coronas very aggressively because it's very hydrophobic. And in the work we did, we found that small polystyrene particles were quite toxic to Daphnia, as you did. But similar sized particles made from more polar polymers were not toxic at all in 72-hour immobilization assays anyway. So I think there are some really important and subtle differences here dependent on the type and nature and surface properties of the plastic. And that's obviously an enormously complicated question when you get into environmental samples, because we don't know where it's been, we don't know how oxidized it is, we don't know what's on it. And the honest truth is, I have no idea how we should go about making toxicological assessments because we know so little about the plastics.
Andrews, I think you are right. You are right, and I think that's why there is also very few studies, in toxicological study, because there are so many factors that are present out there that you find difficult. You have a plastic, you don't know the source, you don't know the type. So many factors are responsible, and that's why it seems that the toxicological aspect of this work is dragging. But we just have to figure out how we have to go about this if we truly want to understand the effects of microplastics on our health and also on the environment. So I want to believe that the more we conduct more studies, the more we get light about the behavior of these materials.
Yeah, I think that's absolutely right. We know far too little currently, and the more of these kind of detailed mechanistic studies we have available, the better the chances are that we can make some kind of sense out of it in a more broader environmental context. So yeah, it's really important, I think.
Yeah, thank you.
Thank you.
Yeah.
So there's one question in the chat from, I think Victor. Victor, please, are you able to unmute yourself and ask the question to Father yourself? I don't know if that's possible.
Yes, it is possible.
Okay, thank you so much.
Did I understood correctly that you found superoxide dismutase copper manganese in the corona, or was that for the gene expression?
You mean this figure? Yeah.
Yes, that one.
This, right?
Yes, that one.
Yeah, yes. I can't understand that question. Can I get your question?
Oh, yes, that you found that protein in the corona, right?
Yes, that this is one of the the proteins that are more highly abundant on the surface of the particle of the nanoplastic, using protometric analysis. So, and based on literature, this is an antioxidant agent. So, which means once it's absorbed on the surface of the particle, it's no longer available for the organism to take care of the free radicals in the system. So as a result, oxidative stress will be induced.
Yeah, I think it's really important that it's like an antioxidant, like it's but you found it in the particle, I guess, after the excretion from the animals.
Yeah, I think it can be, yes. We can actually, we use it to predict what can happen in the physiological system of the animal. If this protein are absorbed on the surface of the animal, then it will not be available for the activities going on in the animal. Yeah.
Thank you. Please, is there any question from anyone for Dari?
Oli is raising up hand. Yeah.
I have
Okay.
I have a question.
Okay.
And I come from a very I don't work with lab-based toxicity studies and things like that, but from a field-based experiment, and I know in other papers have also found that there's, this dual toxicity. It's not just the plastics, it's the plastics and heavy metals, it's the plastics and something else. So for us working in the field, should we also, when we're collecting mussels and things like that, should we actually be collecting other information as well? Because I don't see that often in papers, or maybe I just missed it, but we either report microplastics or we report heavy metals or things like that. Maybe we should be doing these dual studies.
Yeah, I think that's one of the reasons why I have to present this. Is actually relevant in the environment. And I think a group in the Northwestern University in US are actually working in this area. I think in the North Atlantic Ocean, they are collecting plastic particles from the water, from the marine water, and they are trying to subject it to proteometric analysis to see the proteins that are actually absorbed on the surface of the microplastic. And of course, that would definitely give a good information. So I think there are a few groups that are actually looking into this area now in terms of the environmental relevance.
We're still waiting to see if others will have questions for Oluni as well. So I think what I can just ask is that one thing I noticed recently is that most of the chemicals that are, or the plastics that are used for toxicity studies, I don't know whether these chemicals actually come already characterized, whether they tell you which maybe additives they are in there, or if there's other chemicals in there. Because I noticed that some toxicity studies are actually not, the impact of the plastics are not actually from the plastic polymers or the particles themselves, but maybe the chemical makeup or some additives in the plastic that causes the toxicity stuff in the organisms.
Yeah, I think you are right. I think what is important is that what type of toxicity takes place. And that's why we actually use bioimaging to understand. And from us, what we can establish presently is that the immobility of mortality in Daphnia magna when it comes to nanoplastics is as a result of physical toxicity. Because the one that consumed the 1 micron actually expel it, and they are fine. So what you understand with the toxicity of Daphnia magna is physical toxicity. The hydrophobic nature of the particle,
of the particles,
weigh the animal down by sticking into the carapace, and as a result, the animal cannot feed, it cannot do the normal metabolic activity, and that one results in immortality. So, but what you just mentioned about the additives, of course, some of the complaints used to indicate the addictive they add, and most of the time, they either add, additives in order not to allow microorganisms to grow, because most of the particles are suspended in aqueous. So they add additives like sodium azide, to prevent microbial growth. And some of them also add some surfactants in order to keep the particles, in suspense. But one of the things that we normally do in our lab before we use any of these products that we purchase is to first wash them. We wash them severally, centrifuging them. After we've washed them, then we can now be sure that the surfactants that are added and also the preservative that are added are actually not present again before we administer the particles.
But do you characterize them? Like, do you use maybe LCMS or GCMS to try
and look at what is I decided not to include the details there, the details of characterization, because the first thing is we use FTIR, we use SEM, to be able to establish the identity of the particle and to be sure that nothing is present on the surface of the particle before we administer it to the animal.
I think that's one of the things I've been taking notes in review papers these days. Like, people are trying to question, like, most of the toxicity studies in that regards, thinking that maybe the plastic themselves are not really causing anything.
Yeah,
and
because
then the
there's some reports
makeup,
for the 1 micron, we have some report that actually that reported toxicity. So
okay.
It's possible that such toxicity may come from the additives, all the surfactants that are added to it. But in our case, we wash all those particles before we use them. Yeah.
Yeah, that's good. Okay. Yeah. I don't know if
sorry, do you mind if I jump in quickly here? I just had a thought, question, comment type thing. As we move towards trying to standardize and trying to harmonize our efforts to answer these questions, because I think you've raised a really important point, is that we can find as many plastics as we want in the environment, but if we can't tell government and policy makers that they're having an effect, then we're never going to see change. And we actually need to up our efforts on these lab-based studies. But what I guess Andrew was saying with this polystyrene and Elvis with the different, characterizing of the particles and things like that, is maybe there's a way that people who are working on these lab-based studies could have like a document that shares where they bought the I know that is in the papers, but I know that even for us, where we're not using lab-based experiments, but we're using artificial flumes, I mean, we've been able to purchase fibers and make other plastics rather than polystyrene that were much more affordable than we had thought. So sharing that information might be useful for others to access. So maybe that's something we can think about on how we can do that for people who would like to work on these things.
Yeah, that's right. Yeah. Thank you.
Okay. I don't know if others have I think there's okay. Sorry for late. I had to read it. Okay. So recently, I think I saw another paper where they were talking about. I've not read much about fluorescent. Is it fluorescent plastics? Yeah, and then there's I think metal doped plastic. So I realized people are also using plastics where they've doped, like they actually know what they've doped in there, and then they try to trace the doped chemicals in the organisms or something. I mean, maybe that doesn't relate to this, right?
I think what they are trying to do is that, manufacturers do a lot of funny things, in getting their product into the market. So people are also trying to be proactive to see what are the different things that can be brought together, to be presented to the public as plastic. Meanwhile, we may have other contaminants or some of the components that are toxic, to human. And that's what brought about the issue of trying to dope, some of the plastics with metals, and that different studies that are also looking into that.
Yeah. I think I've read papers on that, right? Where they are using metal doped.
Yeah, I think there are imagining studies on that also, where they use the doped microplastics. Yeah. So, and I think one thing that is also important is that we can also be thinking of the models that can be used that are peculiar to the region, the African continent, so that we can also be able to establish standards when it comes to the toxicological study. We want to look at our types of Daphnia. We want to see what is its reaction to these particles. We want to see, for instance, our tilapia fish, which is common to Africa. We want to see the effect of this. And I think these are direct, relevant studies that can actually help us to, in making decisions, when it comes to policy formulation or enlightening people on the effect of plastic pollution in the environment.
Okay. I think one thing I've also been thinking about is because when I started reading the toxicity studies, I was asking myself, what are these concentrations that people use in actually the lab-based work, like the concentrations? Would they actually, or do they actually reflect actual concentrations organisms will be exposed to in the environment? Because what I was thinking was that maybe some of the effects on the organisms are actually from the amount of plastic that are dosed like into the environment of the organism compared to the what I don't think the organisms will be exposed to that same amount.
I think I get what you are saying, but presently we don't have enough analytical facilities to determine what is in the environment. Most of the studies are just predicting the concentration. You cannot really say we have up to this concentration in the environment, or it's just a prediction that we have presently. So until we have until we have enough analytical techniques that can actually determine the real concentration in the environment, I think this predictive study are important. Secondly, when we look at the way we are consuming plastics and it's still degrading in the environment, one day we are going to exceed the threshold. So if that is the case, which means these studies will be a proactive finding for the solutions that may be coming up then. So if we have not gotten this concentration in the environment, if we continue to consume our plastic the way we are consuming them, definitely we are still going to get to this.
Yeah, it makes sense.
The only thing is that the accumulation may not just come once. For instance, the study that estimated the amount of microplastic that is consumed by man per year, about 37,000 microparticles for a person that is drinking bottled water. If such is accumulating in the body, definitely it's going to get to a time that it will definitely result in toxicity.
Yeah, of course, yeah.
And that's excluding the food. I understand that your group just carry out a study, if I'm right, on rice.
Yes.
So when we consume from the rice, we consume from the seafood, we consume from the salts, we may also be arriving at a very high concentration as what is being used in the lab presently.
Yeah.
And that's why laboratory studies are also important in this, so that we can be able to predict what can happen in the nearest future, or what is already happening and we have not actually understand what it is.
I think I understand you, and I think I understand what Andrew is also saying, because concentrations will definitely differ in environment, and their impact or effect on organism also be different as well. I don't know if anyone has a question or a comment or suggestion or something for Danio.
Elvis, I just have one thing to say, and just before we close up, and then just for everyone who's on the call, Sustainable Seas Trust is having their African Marine Waste Conference next year. So I will just share the link if you're interested just before we close up, so that you have that link, but I'll also send it via email on the next communication.
Okay. Thank you, honey. Okay, I think if no one has any question, we'll just thank everyone for spending time with us on our Club Talk 4. I think next month we'll come your way with another Club Talk, which will be the number 5.